It's my pleasure now to introduce Dr. Dung Nguyen, who's going to talk to us about Sarbecoviruses in a UK bat roost.
Good morning, everyone, and thank you for the introduction. I would also like to thank the organizing committee and Miles for the chance to be here and share with you my latest work on the UK bat Sarbecovirus. This is a follow-up to a study previously reported by Jack Crook and others. So I hope you would find something interesting as well. We, as many others, are interested in bats as they serve as the natural reservoir or are postulated as the ultimate source of many important human pathogens, such as MERS or SARS, SARS-1, SARS-2, Nipah virus, Hendra virus, Ebola, and Marburg virus, etc. And it has been estimated that the risk of zoonotic infectious diseases is very high in Asia, like Asian part of China and Southeast Asian country as well, followed by West Africa and West Europe, including the UK. And the current group is carrying out surveillance of viruses in bats in Guinea, in West Africa, and in the UK to understand the future risk of spillover to humans. At the last UK-ICN meeting, I presented this slide showing the detection of coronavirus in Guinean bats in rats, and they include alpha and beta coronaviruses, and they all cluster very closely with previously known bats viruses rather than human viruses. And in this study, today I'm going to talk about the UK bats coronaviruses. The UK is host to 18 bat species, and this show you their names as well as their appearances. And in my study, I have samples from only one bat species, which is the lesser horseshoe bat. Previous study already reported the circulation of coronaviruses in the UK bats. For example, Jack Crook and others discovered the first Sarbecovirus in the UK lesser horseshoe bats, and they named it RHGB01, followed by the discovery of RHGB02 to RHGB06 by APA and colleagues. My apologies that I didn't include your reference in this slide. Another study by Tan and colleagues even demonstrated the presence of a wider range of coronavirus in the UK bats, including alpha coronavirus, MERS-like coronavirus, and Sarbecovirus in lesser and greater horseshoe bats. And among these, only the RHGB7 has been reported to be able to use human ACE2 for infection of human cell lines. In this study, we used 120 bat fecal samples collected in 2024 from a lesser horseshoe bat roost in Liverpool in Gloucestershire, and samples were collected by placing sterile sheets underneath of the bat roost, and any droppings that landed directly on the sheets were collected the following morning and stored directly immediately in DNA RNA shield until RNA extraction. And RNA was screened. The extracted RNA was screened using real-time RT-PCR assay that targets the E genes that previously reported by Coleman and others. And in this method, they also recommend to use RDRP genes for confirmation as well. Screening show extremely high prevalence of Sarbecovirus in samples collected in both June and July 2024 with a wide range of CT value. And this could be explained by the increase in the bat density or in the behavior, social behavior in the maternity colonies, and also in the number of juveniles that are immunologically vulnerable during this period of the year. And I didn't perform the confirmatory test with the RDP genes, but went straight into sequencing to understand which viruses, coronaviruses they were. And we use CISPR, a combination of CISPR and Illumina sequencing on 21 samples that have the lowest CT values. And we obtain complete or near complete genome sequences from all of them. And this OF1A maximum likelihood tree show that they in red cluster closely with the HGB01 previously reported from the same path roots. And they're very highly identical to this virus sequence. And the RBD region is extremely important, and we are interested in this region because it's required for virus entry into cells. And in this region, the obtained sequence differed by 4 to 8 amino acid compared to the HGB1 and HGB7. Again, just to remind that only HGB7 so far has been shown to be able to use human ACE2. I then use AlphaFold3 to predict the interaction between ACE2 of different hosts and with the RBD sequence from different Sarbecovirus. And I just show here some examples. And this, so just to make clear that I use AlphaFold3 on the Google server, and it's quite quick and user-friendly. But the disadvantage is that Google is concerned about the biosecurity of, so they didn't include any viral protein database in AlphaFold3 on their server. So the prediction may be less confident. Despite the lack of the viral protein database, Google AlphaFold3 still confidently predicted the structure of ACE2 and the RBD as indicated by the colors of this structure. And generally the interface predicted template modeling of the UK is less confidently predicted than that of SARS or SARS2 RBD with humans ACE2. And despite this lower than expected, the PA plot, which measure the accuracy of the predicted relative position of ACE2 and one of the RBD, is still confidently predicted potential interaction between one of the RBD and human ACE2, as indicated by the blue color, so the green color in this rectangle. And surprisingly, it also predicted the absence of interaction of the RBD with ACE2 of the lesser hoshibats, despite although this sequence was detected from this species. Because as I said, because of the lack of the viral protein database in AlphaFold3 on the server, I'm performing additional, I'm using more prediction tool to validate the findings of Google AlphaFold3, including local AlphaFold and squid modeling. I'm also performing additional work to study the genetic potential of these viruses, including production of serotype Sarbecovirus with a recombinant spike protein. This method was previously reported by LabCo, in which the RBD of the SARS-CoV-1 was replaced by the RBD sequence from the novel coronavirus. I'm also trying to isolate the UK bat coronaviruses, but so far I have failed. It seems to be difficult to isolate bat coronavirus, so I'm thinking about reverse genetic as well. But this morning, when I had a quick chat with Andrew Davidson, he gave me fantastic advice that we can just extract the RNA directly into cells, and hopefully we can rescue the virus directly from there instead of reverse genetic. So that's what I will try when I'm back to Oxford. So to summarize, we demonstrated high prevalence of Sarbecovirus RNA in UK bat roost, and the identified coronavirus sequence was highly similar to the known Sarbecovirus previously reported from the UK, and differ only by 48 amino acids in diabetes. And we are testing we are evaluating the genetic potential of these virus. I'd like to thank all people involved in this slide, including people from Oxford University, my supervisor, and my colleagues, and also Danielle from the Animal Ecology and Wildlife Consultants, and the FDA for funding. And thank you for listening.
Thank you so much for this presentation. I think we have time for like 1 or 2 questions. Right up front.
Thanks so much for the talk. Do you see any evidence infection because you're sampling the fecal matter from the whole roost? If you did that in some caves in other parts of the world, you might find lots of different viruses. See any evidence?
Yeah, so I try. We use CISPR and Illumina sensing, but so far I only found Sarbecovirus, only one strain in each sample. Yeah.
Let me ask one then. So you reported a interestingly very high prevalence of infection in the material that you sampled. Do you think this is mirrored elsewhere in lesser horseshoe bat colonies around all of Britain, or do you think this is a particularly infected colony compared to the rest? Has any comparable work been done?
Yeah, thank you. So I attended the UK-ICN, and there's a talk from APHA, I think, and they presented quite low prevalence of the Sarbecovirus in lesser horseshoe bats. They found only one sample possibly. I don't remember exactly from how many samples, but it's much lower. So I think, as I explained, there's some reason why, but I think the difference from that study is that the samples was not from the bat roots, but from people who submitted to send APHA the samples. I don't have data from other bat roots, but I remember one study by ALPA and colleagues, and they reported very high prevalence as well, something like more than 40%. And in the same bat roots that I got samples from in previous year, like 2021, 22, when samples were collected in June, the prevalence was quite high as well. It reached 62% in 2023.
Thank you.
Thank you very much.
Thank you.